Professor Brinton here with episode 5 of the note card review. This is going to be a slightly longer video than usual. I have 2 note cards that I'll be dealing with. First is membrane transport. There are 3 basic things that we've discussed with any transport of solutes, which are represented here by these little circles, moving those solutes across a membrane. You have 2 types of passive transport of these molecules, and then you have active transport, as well as bulk transport, which we won't really discuss here. Passive transport is the first one to deal with, and this is considering molecules, in this case solutes, moving directly across a membrane. This is going to occur from high concentration to low concentration. So this is with the gradient, okay? And that means that these molecules, these solutes, are moving across the membrane, balancing out the gradient, moving toward where our lower concentration is. This is considered with the gradient here. So moving from high to low, passive. Passive transport can also require facilitation, which is effectively just requiring a little bit of help. In order to move certain types of solutes across the membrane, you have to have a protein channel. Often, however, these protein channels are simply open, and the solute is able to pass directly through. Active transport is going to be doing the exact opposite of everything that we just discussed. Active transport is not only going to require particular types of proteins in the cell membrane, it's also going to require energy in the form of ATP. So the other difference that is essential that you get between active and passive transport is the direction of the gradient. Here we still have the same situation with a high concentration and a low concentration, but active transport moves solutes against the gradient. So we'll be taking these here from the lower concentration through our protein over to where we already have a higher concentration of these solutes. So with passive transport, either facilitated or just general, you're going from high to low with the gradient. Active transport, you need to add ATP here at this particular protein in order to pile up additional molecules on the opposite side of the membrane. The other thing that's essential here is understanding what types of molecules tend to need each type of transport. Certain molecules move through the phospholipid bilayer in cell membranes quite readily. In general, these are going to be very small molecules and ones that are nonpolar, so they interact perfectly well with the tails that are present here inside all of the cell membranes. So just as a reminder, is what we're dealing with here for cell membranes. We have your polar heads on the outside and on the inside, and then off of each of these polar heads you're going to have 2 nonpolar tails coming in to the middle, and that creates a polar and a nonpolar region on the inside and outside of the membrane. So we've got polar out here, nonpolar in the middle. So small nonpolar molecules such as O2, N2 are going to be able to move readily straight through the membrane very fast. Next up, besides that, are going to be molecules that are also still very small but are polar. Water is the perfect example for this. It's small enough that it is able to work its way through the membrane, but it does so slowly because it's a fairly polar molecule. Up above that, you have molecules that are large and polar, or nonpolar in this case. It doesn't really matter, but if they're big enough that they can't move into the membrane very easily, such as sugar, for example, then they're simply not going to cross the membrane via these methods. They will have to go through a channel. So we move from direct simple passive transport to facilitated passive transport, or to active transport, depending on which way you're moving as far as the gradient is concerned. Beyond that, if you get to either molecules that are simply too large to deal with at a particular protein, or those that simply are not ever going to be able to pass through this nonpolar region of the cell membrane, you're moving on to specific types of active transport or bulk transport. endocytosis and exocytosis. Examples of this type of molecule are going to include very large things such as functional proteins and ions. So, for example, sodium-potassium pumps function in order to move those two highly charged ions back and forth across a cell membrane. That actually brings us to our next half of this, which is dealing with the issues of tonicity and how all of these ways to move solutes back and forth across a membrane allow us to create and use membrane potentials. So hyper means high, hypo means low, and iso means same or balanced. Anytime you need to determine which of these three situations you're dealing with, you need to compare within and without for your situation. Here I have two groups. So I have a cell here in a solution in the purple region that have different concentrations of solutes. Remember, concentration represents a density, so number of particles in a particular volume of solution. So we have cell here and our solution. The cell obviously has a much lower concentration than the surrounding material. So if we compare these two, what we need to do is determine whether the surrounding solution is hypertonic, hypotonic, or isotonic. This is fairly straightforward if you remember that that is the perspective from which you are looking at this. Is the solution hypertonic compared to the cell? Is the solution hypotonic? So in this case, our solution has a higher concentration, so it is a hypertonic solution. If the concentrations were equal, of course, it would be isotonic. So this is always a comparison. How all of this is connected to membrane potential is that we are constantly changing the concentration of ions on either side of a membrane. If we move these solutes across the membrane here via passive transport, then we are increasing the concentration on the bottom side of the membrane. If we have an active pump, say pumping protons or sodium and potassium ions across the membrane, then we are increasing the concentration on this side, the top side of the membrane here. What this does is it creates basically a giant pile of molecules behind a dam. You're building up an increasingly high concentration on this side of the molecule using ATP at an active transport pump. What happens afterward depends on what else is available in the membrane. Okay, added some notes there. So we're crossing the membrane in both cases here, actively building up a charge on the top side of this membrane using ATP, actively at a pump. Elsewhere in the membrane, you have an open channel which the solutes can freely flow through. This is like water running through a dam. So uphill, you are building up this charge, building up the pile of solutes. It flows through, and this is used to actually accomplish work. We've discussed this fairly extensively when we discussed cellular respiration and the ATP synthase pump in the mitochondrial membrane. What we were dealing with there using electron transport chains was a way to constantly build up additional hydrogen ion concentration on one side of the membrane. Then at the ATP synthase on the other edge, you had a passive channel that allowed those hydrogen ions to move back through, actually running the pump, generating ATP here. That's not the only way that this can be used. Membrane potentials are used in a plethora of biological systems. How this is connected to tonicity is the link between concentration and membrane potential. So if you were to consider this cell here with its own little cell membrane, its low concentration inside, what we have is the same relationship that I was just illustrating. You could have in this cell membrane both active and passive transport locations. And let's make this blue one here our active transport. So this one would require ATP. And that's going to be shipping out these solutes against the gradient, piling them up on the outside. If elsewhere in that same cell membrane we have other channels that passively permit these same types of ions through, then they're going to automatically be moving in with absolutely zero effort on the cell's part. Once these channels are in operation, they simply have to be maintained and replaced if they're damaged, but no energy is required in order for these solutes to move through. The result is a constant buildup of these solutes on opposite sides of the membrane. You're creating a battery where you have a charge building up using a bit of energy, and then elsewhere that charge is released, actually doing work. So tonicity is related to membrane potentials because you're going to be constantly creating hypertonic or hypotonic environments for this particular cell or for certain regions of the cell in order to facilitate the hydrogen ions or other types of ions actually doing work for the cell.